We are live. Gilberto, do you want to start for us, please?
Yes, please. Thank you, Thiago. Obrigado, Thiago. Eu queria refazer os announcements que eu fiz no último seminário sobre as eleições da sociedade. Já está aberta, repito, a temporada de caça à nova diretoria. A gente tem que... Vocês devem ter recebido o correio, foi despachado para todos os sócios que estão cadastrados, e será a apresentação de uma chapa única, ou chapas, né? Quem quiser concorrer. Mas será concorrência por chapa, não por cargo. Então, a chapa teria candidato à presidência, vice-presidência, tesoureiro, secretário-geral e um secretário que é da cidade do Rio de Janeiro para tomar conta da tramitação, né, que a sede da sociedade aqui, como faz a bioquímica, como faz as sociedades normalmente. Então, eu queria chamar a atenção de vocês. É muito importante, perdão, a gente está incentivando muitos jovens a entrarem nesse jogo. Nós precisamos renovar a sociedade. Não tem mais sentido continuar aqui no Rio do jeito que tem sido esses anos. Eu preciso de sangue novo, uma endogenia muito grande aí, novas ideias, nova flora, nova fauna. Candidatem-se, montem uma chapa, apresentem a chapa. Vocês têm 30 dias para isso. Depois a gente vai ter uma eleição remota, vai ter uma semana quase para a eleição, e depois, 30 dias depois, 4 semanas depois, a gente vai ter uma Assembleia Geral, já está convocada, as datas todas foram divulgadas, para a gente fazer, então, a homologação e a prestação de contas e os etc. da diretoria que está saindo. Ok? Então, incentivado fortemente a vocês apresentarem chapas. Várias chapas, concorrência, isso é ideal, isso. A gente vai ter um congresso ano que vem, junto com a BR+, então vai ser um Joint Congress, ibero-americano, e então a gente precisa de sangue forte, novo aí, para montar esse congresso. As perspectivas são excelentes, montar muita coisa em proteômica aí para espalhar esse evangelho proteômico, como eu gosto de dizer. É isso, Thiago. Obrigado. No final, eu gostaria de repetir essas mesmas palavras, mais sucintamente, para quem tiver entrado durante a programação. And hi, Ole. That's with you and Thiago now. Thank you.
Só lembrar também, pessoal, que no final da sessão, nós vamos ter também a premiação, né? Nós vamos divulgar os premiados na sessão dos Student Sections e no Young Investigator Section. Então, por favor, permaneçam nessa sessão após a palestra do Ole, tá? So now I will change gear to English. So it's a great honor for me to introduce Ole. So Ole, he's a professor at the University of São Paulo, Denmark, and he's a group leader in the protein research group. Ole, in the beginning of his career, he is one of the key scientists in the field of proteomics and mass spectrometry. So in the beginning of the 2000s, end of the 90s, he was very focused in developing new strategies to study post-translational modifications. So Ole was really involved in developing phosphate enrichment methodologies and so on. So after a while, let's say in the last 10 years, if I'm not wrong, he changed a little bit focus for the middle-down and top-down proteomics. So he's studying a lot of histone modifications. And I've learned recently that also he's very interested in imaging mass spectrometry to study cancer. And yeah, I think he's going to show us a lot of really exciting data. And it's also important to mention that he published hundreds of papers, won several prizes and awards as the Elite Force 2009 prize, Juan Pablo Albar Proteomics Pioneer Award in 2019, the MCP Lectureship Award. And yeah, he's been he was also involved in a lot of administrative things, as he was already he is the current the director of the Willem Center for Bioanalytical Science, and also the PrO and MASSA, the Danish National Mass Spectrometry Platform for Functional proteomics. And yeah, without further ado, I'd like to give the word for you, Ole. And again, thank you very much in the name of the Proteomics Society, Brazilian Proteomics Society, for accepting to give this talk. And this is not a coincidence that the last day, the last webinar, it's your time. Thank you very much.
Thank you so much, Thiago, for this very nice and kind introduction. And I just want to make sure the sound is okay now, and you can all hear me. Yes, and then if we switch to my slides, then I given a title that is rather broad about proteoform diversity and how we can use middle-down and top-down proteomics to study these proteins. And Thiago, I cannot see any of you now. I can only see my own presentation. So please interrupt if there are any issues that we need to discuss along the way.
No problem.
Yeah. Okay, so as Thiago just mentioned, that I've been involved in proteomics for many years. And actually, when I was preparing this talk and another talk during the last week, I realized that it's exactly 25 years ago that this particular paper came out in PNAS. Back in December 1996. And this was really one of the first papers showing that we could use mass spectrometry to measure proteins, in this case by peptide mass mapping, by multi-mass spectrometry, and also by sequencing peptides by tandem mass spectrometry using nanoelectrospray, MSMS, that we could assign peptides and proteins to genomic databases, and this was applied to yeast back in the days. This was the year that the yeast genome was published, before the human genome was known. So this was really very early on, and it also reminds me that I'm getting older now, of course. So 25 years of proteomics, and I think it's still true what was said in this abstract, that this study established that mass spectrometry provides the features and the capability to identify proteins to connect the genome to the proteome. So I think it's pretty interesting and fascinating to think about all the technology development that has happened over those last 25 years and compared to where we are today. So my research program currently looks like this, so I try to make an overview here, what I'm interested in. So on the one hand, I'm interested in mass spectrometry as a technology and to develop methods around mass spectrometry to measure proteins and peptides. So that has to do with quantitative proteomics, looking at post-translational modifications of proteins. And as Thiago mentioned, I'm moving into middle-down and top-down mass spectrometry, and I'll give you some examples today. Also, I find ion mobility spectrometry rather fascinating, so I'll also touch on that. And of course, for all we do, we need informatics approaches. So proteomics informatics is sort of going through all my slides today and all my projects. So I'm also very much interested in chromatin biology, protein DNA interactions in the context of aging and cancer. Again, focus on modifications of proteins, particularly histones and all the many proteoforms of histones, and how these are involved in both transcriptional regulation and epigenetic regulation of our genes. And also in the context of cancer, in the last few years, we have focused more and more on kidney cancer, together with pathologists and oncologists at the Odense University Hospital. And then in the last few years also, I started establishing mass spectrometry imaging. We acquired some instrumentation so that now we can actually obtain mass spec images from tissue sections. And we just recently installed a new instrument where we can do MALDI-2, that is post-ionization. And we're pretty excited to see how that's going to affect our results. So I'm not going to talk about imaging today. We only have one paper out from last year, and you can have a look at that if you're interested, how we can use mass spectrometry together with proteomics and mass spectrometry imaging to study kidney cancer tumors and classify different tumors. So to run this research program, I'm fortunate to be directing a number of infrastructures here. So we have some advanced mass spectrometry provided by the Willum Foundation and the Novo Nordisk Foundation and the Danish government, who gave us, or actually awarded some grant and funding for acquiring some of the most advanced mass spectrometry. So I'll show you some of the results today using the advanced mass spectrometers that we have. So the Yes, so my fascination of post-translational modifications goes back, almost 25 years, because it turns out that post-translational modifications, of course, regulates many of protein functions, especially the activity of proteins and the location of proteins. And this is very difficult to study by genomics methods, so we basically need to turn to proteomics to really measure what the protein, where they are, what they're doing, and how the interactions are with proteins. So we have worked with this modification-specific proteomics concept for many years now, where we combine PTM enrichment methods with mass spectrometry to go deep into the mapping the modifications and quantifying modifications in proteins. And we can today, of course, identify and quantify thousands of phosphorylation sites and methylation sites and glycosylation sites in proteomics experiments. But usually, in many of these types of experiments, only one type of PTM is considered, so usually phosphoproteins, for example. But what turns out, of course, is that each protein can contain different types of modifications that actually cooperate. So methylation and phosphorylation can co-regulate functions, and also acetylation, ubiquitination, and so on occur together to provide different functions in proteins. And basically every protein, at least in mammals, is modified in some way, and usually by multiple modifications. So we really need to focus in on developing methods where we can study these coexisting modifications, coexisting PTMs, and to investigate their biological functions. And so the workflows we usually follow is to prepare the sample. Either it could be a tissue lysate or a whole cell lysate, or we can go into extracting particular protein complexes or organelles like mitochondria or nuclei. And then we will work on the enriched samples here usually by electrospray, but MALDI can also be used sometimes. And then we sequence these peptides and map out where the modifications are present. And of course, we can use quantification methods, either label-free or stable isotope labeling methods like TMT, to study the differences in modifications at different time points or by different perturbations, dose response of drugs, for example, or hormones. And then we can, hopefully in the end, assign some interesting functions to the proteins. Okay, I just need... Yes, here we go. So as Tiago mentioned, I've worked in this field for a long time now, and we started out in the early 2000s working a lot on phosphoproteomics and actually also on GPI-anchored proteins. So these were the early days where we were happy if we could just identify, 10 or 20 phosphorylation sites or a few hundred phosphorylation sites. And of course, technology has moved on. And so now today we can get, thousands of phosphorylation sites in proteins. We then moved into quantitative phosphoproteomics using SciLag at that time. Now more and more is going into the direction of TMT-based methods. But still the concept of using affinity labeling, for example, by IMAG or by titanium dioxide, and using Orbitrap technology, if we transform mass spectrometry, back in the day it was FT-ICR, which is of course going a little bit out of fashion now, in at least in proteomics, and now the Orbitraps are basically ruling the world in many labs. But the overall concepts are still holding through today, and then much of this technology is still being developed and scaled up, so we can now study many hundreds of samples and compare them to each other, for example, in clinical proteomics studies. So in the last 10 years or so, I moved a little bit away from the, let's say, fundamental phosphoproteomics and modifications, and moved more into the studying the combinatorial modifications in histones. And this is where we started working on the middle down strategy, to study big peptides, usually 5 kilodalton peptides from histones. And we have applied this to study mice, and I'm going to go a little bit more into detail with this particular story that is shown here. That was published in 2017 in Nucleic Acids Research, just to illustrate the concept. And from there, I will then move more into ion mobility spectrometry and also some top-down mass spectrometry for protein analysis. So why is it we want to study these biological systems by proteomics? That is, of course, because there is a big drive right now to go into the direction of precision medicine. We want to stratify patient groups and understand better how different patient groups respond to drugs and how they respond to disease, of course, and how the prognosis are. And so it's really important to map out the whole genome, of course, but then also more phenotype type features. And this is where the proteome comes into play, but of course also the metabolome and the epigenome are very interesting in these concepts. When we study the proteome, we can begin to learn about the signaling networks and regulatory mechanisms, and also interactions in the cell and in tissues. You know, which proteins interact with which, or which genes interact with certain proteins. So my interest is, as I said, is to study chromatin, to get a fundamental understanding of what governs and regulate protein-DNA interactions in the context of transcription, gene regulation, and also epigenetics. And we worked in aging a bit, and then more and more also in cancer. So basically, we want to ask the question: how do these different post-translational modifications of proteins turn on and off the activity of particular genes? And all this is mediated by transcription factors and epigenetic enzymes, but also of the histones that are actually opening and closing the DNA. And that's what is shown in this slide, where I try to give a little bit of an overview of how chromatin proteomics, if we can call that a field, how we can use this to study protein-DNA interactions in chromatin. So of course we can identify chromatin binding proteins by doing immunoprecipitation or isolating promoter regions or different genomic loci. We can tag particular proteins and pull them out and look at interactors and so on. So there's a lot of activity in this field here, and it's being embraced by many cell biology labs and epigenetics research labs to use mass spectrometry and proteomics for this purpose. We can also, of course, study the modifications in proteins here to learn more about the regulation and the dynamics of chromatin. And here the proteoform comes into play. You know that proteins can exist in different states depending on the number of modifications and types of modifications and the combinations of modifications. So we're very much interested in mapping out the different proteoforms to understand what we call proteoform families, because it's a very diverse set of proteins, for example, in the histone family. And then, of course, we would like to understand also the interactions between proteins and DNA and between proteins, and what stabilizes them and what keeps them flexible. And then we can begin to build on to do more advanced studies using time course analysis, do dose response by adding hormones or growth factors or drugs. We can knock out particular factors like a transcription factor and then see what happens to the proteome or the genome. And of course, we're now moving more and more into clinical samples as well. We have an ongoing project now where we are involved. We're looking at cancer tumors and studying the histones in cancer tumors to try to figure out what's going on with the regulation of the genome by looking at the histones themselves. So over on the right side here, you can see the nucleosome, that is the protein complex around which the DNA is wrapped. And it consists of the 4 different histones, 2 of each, that forms the nucleosome core. And then the N-terminal tails of the histone sticks out. And these can then be, they serve like a scaffold to recruit other proteins, including enzymes that can write or erase or read the signals in the histone tails. So these are the transcriptional enzymes or epigenetic enzymes that work in collaboration with the histones to open and close the chromatin. And this is shown down here, how the modification status of the histone here, for example, histone H3, if it's acetylated here in the tail sticking out, this region here, then if that happens in a number of nucleosomes in a loci in the genome, then it will open and the DNA will be more accessible for transcription, for example. Could also be repair or recombination. And in the other case, it's repressed, and then the acetylation is exchanged for methylation that then leads to compaction of the DNA. So this is a sort of a simple description of how these modifications on histones are really, really important for regulating the gene activity in that loci in the genome. So this is a bit more detail here, where we look at the nucleosome and the DNA wrapped around it and the tail sticking out. And here we have many arginines and lysines that can be methylated, and all the lysines can also be acetylated, and there's some serines as well that are phosphorylated. And on top of that, we can also have ADP ribosylation and ubicylation and so on. So there can be a lot of sites here that can be modified in these histone tails. And mostly when we do analysis in bulk, that is the global analysis of histones from cellular nuclei, we'll see lots of methylation and acetylation. And remember that methylation can be mono, di, or trimethylation as well. And any of these sites here. So there are many combinations of modifications. So by imagining all the modifications you can have here, you end up with hundreds of thousands of combinations. And in effect, we see some hundreds, maybe 1,000 different combinations here when we measure by mass spec technology, as I'll show you briefly. And here you can see an electron micrograph. We actually can see the open and the closed states of the chromatin here in the micrograph very clearly. And again, depending on the combinations of modifications here, usually many acetylations will lead to transcriptional activation, so it's on state. Whereas if many of these lysines at K27 and K9 and K23, for example, when they're methylated, then it's an off signal, and in this case, they are trimethylated. So again, modifications are really important. So initially, when we started this, we used the standard bottom-up proteomics strategy to analyze triptych digests of histones, and it actually worked out very nicely back in 15 years ago when we started this, but it quickly turned out that the methylation and acetylation of the lysine residues really complicates the interpretation of the spectra. So that's why we moved a bit more than 10 years ago to the middle down approach, where we cut a big part of the tail. So we have a 50 amino acid tail that we cut off using a glue C or a SPEN, depending on the histone type. So now we have larger peptides that will have multiple modifications on them. And now we can begin to study these combinations of modifications. And these would normally be lost in the triptych digest over here using the bottom-up strategy, because each individual modification would end up in a particular peptide, and you couldn't study the combination anymore, at least only in a few cases then. Whereas the middle down will give us all the N-terminal modifications. And now we are moving towards the top-down approach, where we take the whole proteins and will analyze them inside the mass spectrometer, and then we can map all the modifications and monitor all the modifications simultaneously. I'm not going to show any work on top-down analysis of histones today, but I'll show some other examples on top-down protein analysis later on. So for the next few slides, I'll go more into the middle-down proteomics approach. So basically, we want to study the roles of co-occurring modifications in proteins, and this is a focus on histone H3 in mouse tissue. And this is a published study that I mentioned before. And then from there, I want to go more technical and move into how we can use mobility spectrometry to study near identical or isomeric histone peptides. And these are the N tails, as we call them, the N-terminal tails, which are 50 amino acid long, so on the order of 6 kilodalton inside. So for the middle down analysis, we cleave the histones with glue C. And I should just say that we usually, the sample preparation is extraction of the nuclei from cells or tissues, and then we isolate the histones by an acid precipitation, and the histones will be in the soluble fraction, and then we can digest them, and we don't separate the histones out. We treat the whole histone mixture of the 4 different types and all the proteoforms within those histone families, and then we look at the bulk histone preparation and treat it with glue C in this case, and then we will focus on the H3 tail usually, at least in the studies I'm going to show you today. So we have this 50 amino acid tail here that can carry these different modifications that I mentioned before. So we'll see many different modifications and combinations of modifications. And usually we will measure more than 1,000 different modification states within that one peptide. So you can think of this as one sequence that occurs in hundreds and even more than 1,000 different states because of the different combinations of modifications. So the challenge for mass spectrometry, of course, is to be able to distinguish all these from each other, and this is why we use LC to separate out to some degree the different modified proteoforms, and then we will use tandem mass spectrometry to sequence them and map out exactly where the modifications are. So when we do that, we can actually begin to catch these combinations of modifications, and we can determine the relative abundance of them. So here I just listed a few of them. So here you can see the K27 dimethylation together with the K36 dimethylation, which is very abundant, which is known. And then other combinations are less abundant, but we see many of them. And this list would continue, for hundreds of different combinations here. We can also distinguish sequence variants. So there's the H3 histone comes in the H31 or H32 variant that can be distinguished from the H33 variant because there's one alanine and serine that is changed between the two species. So by sequencing through this region where we have the alanine or serine in this region, we can actually tell which one is which. So we can tell which proteoform, which sequence variant is actually modified and which modifications are carried on each of them. So as you can see in here, we can see the H33 form carries many more modifications than the H31 and 2 forms. And then, of course, we can do site-specific quantitation as well, looking at lysine 4 and see what modifications are there, and we can do it in a relative manner. So here you see that lysine 27 is heavily occupied by modifications. Okay, so this is just a brief outline of the method. So we have all these histone tails, a very complex mixture. We separate them with a weak cation exchange helic. It's a method published by Ben Garcia's lab some years ago. And we adapted it and optimized it also with using a pre-column. That was the idea of Simone Sidoli, who published this paper back in 2014 with our optimized method. And then we also adapt the ETD MSMS because the electron transfer dissociation is a very efficient way to sequence large peptides with multiple charges on them. And this is, of course, a work by Don Hunt and colleagues who pioneered the ETD method back in the 2000s. So what we get from this, the helix separation, is that we can separate out the different proteoforms of the histone H3 in this case. So you see here the late eluting peaks are the ones with no acetylation, and then there's a group here with one acetylation and another group with 2 acetylations. So the more acetylation, the earlier it elutes from the hydrophilic interaction column. And if we then go and look into this mountain of peaks here, we can actually see that the group of tails, the N-terminal tails here with no acetylation, it splits up further in the number, depending on the number of methylations. So the more methylations, the earlier it elutes within that whole group of peptides here. So we can, based on the chromatography, we get both acetylation information and we can also get some separation of the methylated states. And that helps us then when we sequence the peptides by mass spectrometry, we can much easier assign the modifications because they are rather pure when they enter the mass spectrometer and they don't mix up with each other. So this is just some of the data where we have the mass spectrum here. You see these are highly charged species, 10 charges on this 546.4 M/Z species. So they have a molecular weight of about 5.5 kilodalton. Here we zoom in and see the charge states very clearly. And then the MSMS looks rather messy. As you can see, there's lots of peak here, lots of information, and then we can deconvolute it using Mascot, for example. And here you see the assignment of all the fragment ions in the ETD spectrum, going from about a few hundred M/Z all the way up to 5,000. Again, this is a deconvoluted spectrum created by Mascot. And we get very nice sequence coverage in this peptide. And we cover also this region with the alanine for the H312 and this would be a serine then for the H33. So it's important that we can actually sequence through this region to tell which protoform we have here. And in this case, we can also map a trimethylation and a monomethylation here and a dimethylation down here. So we get very efficient fragmentation of large multiply charged peptide ions. We get very high sequence coverage, and we get usually unambiguous assignment of multiple modifications within these peptides. I'm just going to change the battery in my mouse as we move on here. So the example I want to show you is a project where we study the histones in the context of mouse aging. And so this was a project by Andrei Tragowski, who was a PhD student with me a few years ago. And the idea was to study how these modifications change over time, in this case during aging of the mouse. And the main aim was to demonstrate that we can actually use this middle-down mass spectrometry to study the different histone protoforms from real tissue, in the case mouse tissue. These protoforms, H33 from the H31, and we also wanted to study how different sequence variant could be modified in ways. So it was the study in different people. We isolated liver, kidney, and from time points, 3 months, 5 months, 10 months, 18 and 24 months, and we then went for the histone extracts and analyzed them by middle-down analysis. And I'm going to jump straight to the data. And as I said, this is published, so you can look more into this paper here. So basically, we focus on this N-terminal tail from the histone H3 with the many methylation and acetylation sites. And what you see here on the top row is that the histone H33, that's the one we can distinguish by looking at this alanine to serine variant. You can see it turns more red over time. So the H33 increases over time, and this was known for other tissues, for a number of tissues in chicken and other species. But here we show in the mouse that it's the case that over time, as tissue matures, the H33 protoform takes over from the H31 and versa. The H31 and 2, they're usually used in the early development and growth phases for cells. In the case of cancer, because cancer tumors are proliferating tissues and they have a high level of H31 and 2, but very little H33. So we can actually use the histone protoform as a sort of indicator of the growth state of a tissue or a cell culture. What you see further down is that the modifications we can then follow also. And in the first group here, we have the methylations at different positions in the histone tail, and these are the increasing ones. So we see increases in dimethylation and monomethylation and trimethylation in some sites. The blue region here are those methylation sites that are reduced over time as the mouse is aging. And then we have sort of intermittent here where we have different features showing up for the different tissues. So there are some tissue-specific variations as well. And then below here, we see acetylation is not really changing a lot over time. So basically the take-home message here is that methylation of histones changes a lot during aging in a site-specific manner, whereas acetylation is not changing very much. Plus, as I said, we can see that the H33 increases with age. So already here we've got a very good feeling that we can use this middle-down mass spectrometry method to really learn something about biology and how histones change over time. And you can see here also the reproducibility is really nice, and because the error bars are rather small. So we really can measure these changes even at a few percent level here across time for these mice. Yeah, so now we have measured all these modifications and also combinations. We can make this kind of a logo slide where we can see which modifications are most abundant and which combinations are most abundant. So it's just a nice way to see, on the one hand, that we have a few modifications that are really, really abundant with a big font. And then we have just, you can see the number of combinations we have here is into the hundreds. So it really illustrates very nicely the complexity and the abundance differences between these different modifications. And this is bulk histone, so it's for the whole genome, if you will. So we have not studied yet this single gene histone patterns, but that would be really very nice to do. And for this, we need more targeted analysis using, for example, chip-based methods where we immunoprecipitate particular transcription factors that can bring down histones with them to study modifications. And there are a number of groups around the world working on this as well. So this is just a different way to show the data. Now we have the different tissues here, and now on the right here we have all the combinations that I mentioned in the logo slide before, and this just goes on and on and on. Several hundreds, and you can see some of these modifications and combinations actually are rather change a lot over time, go up and some go down. In the gray bars here in the middle, we have the abundance of them. So you see this one here, the K9 dimethyl, K27 trimethyl is very, very abundant over time, all time points basically, and it shows a decreasing trend for all the tissues here. So you can dive into much of this detail, if you will. So then we defined what we call the PTM interplay, where we, together with Veit Zwemle, developed an algorithm where we could look at the expectation of seeing a particular type of modifications occurring together. So we basically look at the frequency of how often do we observe it and how much would we expect to observe a particular pair of modifications. And here you can see that if you have codependent modifications, for example, K36 dimethyl and K27 dimethyl, they would both occur together, then it would get a line like this, increasing slope. And we see that for all tissues. So these are PTMs that like to occur together and frequently are observed together. Whereas if we have modifications like K36 dimethyl and K27 trimethyl, they rarely occur together, so we get a negative slope. So now we can begin to tell which modifications like each other and which ones hate each other, basically. So we have positive crosstalk or negative crosstalk as shown down here. So for the two dimethylations here, we have positive crosstalk, and for this dimethylation K36 and the dimethylation of K9, we have negative crosstalk. So they rarely occur together. So that's also informative, to understand how genes are regulated. So this was what I wanted to show for this middle down histone analysis, that we have a robust platform for sequencing these peptides, and now we can work with real tissues. And this was sort of the precursor for our current study where we look into human cancer tumors. So we're basically now applying the same methods to study human tumors. So we can distinguish the proteoforms, the sequence variants, and the modifications. And we can find out what types of modifications are relevant in the context of aging in this case, and we hope soon to also see what happens during cancer. And for this, we also had to develop a number of computational tools to identify the PTM crosstalk and interplay, as I just mentioned. And we have a database, and we have a tool called Qtov Mapper that is also available on our website if you're interested. Okay, so now I'm going to move a bit into the technology of ion mobility spectrometry, and that's actually because we saw issues with the middle down strategy that sometimes we do have co-occurring peptides, or peptides that co-elute, and therefore and also have very similar masses, and the MSMS could not resolve these species. So in some cases, you may have 3 peptides, same sequence, but with modifications at different locations. So it could be a methyl in 3 different sites, and they, of course, would have the same molecular weight, and you would need somehow to resolve these species from each other. And when you have more than 2 peptides in one MSMS spectrum, 2 same mass peptides, or basically 3 same mass peptides, 3 isomers, you cannot really resolve them by MSMS, because you will always get a fragment ion that is from anywhere in this peptide sequence when you have this mixture here. So you couldn't tell which is which, basically. So for this, we think that ion mobility could come in and help separate out these gas phase ions. And then after that, we can do MSMS. So instead of having 3 peptides going into the MSMS simultaneously, we would then resolve them beforehand and have individual MSMS spectra of the 3 species. And this is where ion mobility could play a role. So ion mobility separates molecules based on interaction with a gas as they move through a field. So in this case, we have ions entering from left to right in the electric field, and the drift gas going against here. So the ions will see a strong counter force that will slow down the bigger ions. There's a bigger, what is called collision cross-section, a bigger surface area here. So the big ions will see more resistance than the small ions. And just a small note, those of you who are familiar with the Qtov instrument from Bruker, this is reversed. So actually the ions are brought through by the gas, and then they see an opposing electric field, and that gives the separation by ion mobility. But there are different implementations of this technology. And yet another implementation is FAMES, field asymmetric waveform ion mobility spectrometry. And this is what is called differential ion mobility. And here a voltage waveform is used to stabilize or destabilize ion trajectories. So in this case, with this particular setting of the voltages or the amplitudes here and the waveform, the green ion will be allowed to go through, whereas the red ions would be deflected. And then by changing the amplitude here, you can then allow one of the red ions to go through instead of the green ones, for example. So this is a CV, this voltage here, that is compensation voltage that is used to filter for particular ion species here. So actually we started working on this about 5 years ago with Alex Schwarzburg from Wichita State University in the US. And for this purpose, we wanted to synthesize histone tail peptides where we could put the modifications in the different places. So for example, for the acetylation, we would have one peptide with acetylation in K9, another sample of peptides with K14 acetylation, and another again with K18 acetylation. So all the peptides would carry one acetylation, but at different positions. So they will all have the same mass. They are isomeric and only differing by the position of the modification. And we did that for phospho and trimethyl and monomethyl peptides as well. And then we tested this with a FAMES device that is developed by Alex Schwarzburg here on the right in the photo, next to Pavel Sliaha, who was a postdoc with me. And so he was basically the one driving this project over some years, and very successfully. As you can see, we have a number of papers together. So we mounted this FAMES device on our Lumos instrument and then investigated histones. And this is just one slide from the paper here that we published in Analytical Chemistry, that we look at the histone tail from H33, so it's the 5.5 kilodalton peptide, and all the 3 peptides shown here, they have exactly the same molecular weight, they only differ where the modification is sitting. So here's the K4 monomethyl, the K23 monomethyl, and the K9 monomethyl. And you can see that by using different compensation voltages, we can actually separate out these different species. So the red and the blue and the yellow are completely separated in the gas phase. And you can also see that some of them actually occur in different conformational states. So you have a number of yellow peaks here. So apparently in the gas phase, there are still different conformations that we can separate out by ion mobility. Same goes for the red one. That's very clearly, it has 2 major forms and some minor forms as well. But we can basically separate the major forms for these 3. And this is really amazing because the peptide is 5,500 Daltons, and the modification only has a mass of 14 Daltons, right? So, and depending where we put those 14 Daltons on this 5.5 kilodalton peptide, we can actually separate it out. So it's less than 0.25% of the mass that is used here to actually distinguish the different forms. I really find this fascinating. We did this also with the real samples derived from mouse embryonic stem cells with the Helix LCMS method that I just mentioned. And here we put the FAMES in front. This is now a circular FAMES, not the planar FAMES as I showed before. But we can identify more than 500 proteoforms. And again, you can see that we, depending on the computation voltage, either here or here, we get completely different profiles. So here the sample was run multiple times at different CVs. And we get different output, different histone tails are detected. And this is just quickly shown here, where on the upper left here, we have the unmodified species with rather homogeneous trace. But as we move over here with all the trimethylated species, you can begin to see separations. Trimethylation with 1-acetyl also gets separated somehow. And here we have 5 methyl groups sitting on a tail, and they also separate out to some degree. So I think this is a great potential for future of very complex samples where we have these different protoforms. And we are pretty excited to explore this more, both with the FAMES, but also with the TIMS technology on the Qtov systems that we got this year. So during the last few years, then Waters launched a new system. It's a Qtov system where they put in a cyclic ion mobility device. So this is the outline of the Qtov with ion guide here in front, the quadrupole. Normally the ions would fly through and go into the time of flight here with orthogonal acceleration. But now they put a circular or cyclic ion mobility device here. So as the ions fly through, they come here in the red trajectory, then they can be trapped here by an orthogonal field. And this is a wave, basically electric wave, that so the ions will surf along this wave and circulate through here. And you can pass once or twice times or 20 times or more. And the longer the more passes, the better resolving power you get in the ion mobility separation. And then once you're happy with the separation, you can trap the ions again back here. They'll come back down here, and you then orthogonally accelerate them out, and then they fly into the orthogonal TOF, and you can detect the ions. So this is a pretty amazing instrument. And 2 years ago, I visited Waters to analyze some histone tails from this synthetic series that I just mentioned. So this is just a mass spectrum of the 10 plus monomethylated species. And here the arrival time, it takes about 19 milliseconds for one pass around the circular IMS. And we could then do it for the 3 different species with the K4, K9, or K23 methyl, or the mixture of the 3. And you can see here they separate a little bit when you have the individual traces, but when you have them together, it's really hard to deconvolute. But then of course we could just allow it to pass through some more times. And here is just the arrival times after 1, 2, 5, 10, and 20 passes, so up to 155 milliseconds. That's what it takes for 20 passes, so it's still pretty fast, even though they travel 20 times around this about 1 meter cyclic path. So it's like a 20-meter ion mobility device when you use 20 passes. And I should say here that the losses are really small, so we have like 98% transmission efficiency. So you actually maintain your ions very well in this system, even after 20 passes. And so this is the monomethylated K4 species. We get a very nice peak here. And again, you see there's a major species and then a little peak here next to it, or maybe 2, showing that there are different gas phase conformers. And this is with the resolving power of 290, which is very, very good for an ion mobility device. And then when we take all 3 of the isobaric or isomeric species, you can now see that we can separate them out after 20 passes. So they all fly together around the cyclic IMS, and after 20 passes, they have separated, even though they have exactly the same mass at 5.5 kilodalton. and only differ by the position of the methyl group. So this is a mixture of the two, again 20 passes, very nice separation. And then we can eject them from the cyclic IMS and into the collision cell and do CID, and then we can read out the fragments in the time of flight. And here we are looking into the mobilograms. So the drift time is on the y-axis, and if you look along the x-axis here, then each of these groups here corresponds to, you see there are 3 groups basically in your horizontal plane, where you can get sequence ions for each of the 3 different species. And they're actually shown here at the spectra, and you can see they're very similar in some regions, but then there's some differences that tells where the modification is sitting in these. So this really shows that when you have an isomeric, a mixture of 3 isomeric species, you can separate them by mobility and sequence them independently afterwards. We also tried with the trimethylation species after 20 passes, and you can see here we get separation of the K4 trimethylated from the other 2, but the K9 and K23 have some overlap, shown here in the mixture. And of course, we can then ask the system to go for 60 passes instead, and now we have a resolving power of 500, which is very high fine mobility, and now we begin to separate the 2. And even though they're not fully separated, we can do MSMS of these, and when you look again in the mobilogram, you have 2 clear distributions of ions here that you certainly can discriminate between the 2 species. So this is pretty exciting. It's a very advanced and specialized instrument, and it cannot yet do LC-MS in this mode, but it allows us to do fun experiments to separate out these isomers. So I see a bright future for mobility also in the middle down world, where we can add specificity to molecular characterization using the collision cross-section. We can separate these same mass or isomeric protoforms, and we can also see gas phase conformers, so we have multiple signals from one species. So FAMES can be used for this, as I mentioned, but also and also this can be applied to real biological samples of entails of histones from embryonic stem cells. And then these early results on the cyclic IMS also shows a lot of promise for looking at very, very similar or near identical histone tails, and of course can be used for many other types of biomolecules. Okay, so for the last few minutes here, I want to go to the right side here to the top-down proteomics and just show you some recent results we got when we studied intact proteins. And I like to use this as an introduction to top-down proteomics. You shouldn't try this at home. In the lab, we had a mosquito that actually got trapped in the inlet of the mass spectrometer. This is real top-down proteomics. If you can put the whole insect into the mass spectrometer and sequence the proteins, it didn't work. But the mosquito died in the process, first by electrocution and then by being sucked into the vacuum. And my students caught it and took a nice photo of it. So it's pretty cool. So try to keep insects out of your laboratory. So what I want to talk about is this top-downer tool that we developed over the last few years. And the idea is to have an automated method where we could scan through many, many different instrument settings in a short time to find out what are the best fragmentation settings for a particular protein. So we use the Orbitrap Lumos, and more recently also the Eclipse, that allows us to do CID, HCD, ETD, and UVPD systematically. Of course, if you have to do this manually, you would have to sit in front of the instrument for many hours and change parameters and settings. So what we developed was a methods generator program that could generate all the methods beforehand, guided by the user, of course. And then it would transfer the methods to the mass spectrometer that then would, over some hours, would scan through all these different settings and acquire the data. And then we could read out the data, and this is one of the readouts here, where the color and the size of the balls or the circles here tells how efficient it was for getting a good ion intensity for a particular setting. So this is just the parameter space. And we also have this readout where we can look at the sequence coverage. So the amino acid sequence of the protein is here on the y-axis, and on the x-axis we have all the different settings. So CID, HCD, ETD and ETCID and ETHCD and so on. So just by glancing at this map here, we can see which method really works and which does not. I'm going to go through this in more detail. So what we want for comprehensive protein characterization is the intact mass, of course. Find out how heterogeneous is this sample. Do we get one major species, or do we see many proteoforms or oxidized products or deamidated products and so on? That's important for the pharma industry. We want then to get efficient amino acid sequence, optimally 100% amino acid sequence coverage for verifying the sequence and to assign modifications. And of course, my interest would be then to look at coexisting modifications, and this is where the histones would come in. We're not there yet, but I'm just showing you maybe a first step on the way. And we also want to monitor clipping and processing, of course, and other modifications. And eventually, we would like to do real top-down proteomics and to quantify the proteins and the modifications. So we're not there yet, but it's a field that's moving very fast, and there are really nice papers coming out these years from different groups around the world. So it's really interesting to be in this emerging field of top-down proteomics. So we use this advanced mass spectrometer, the Lumos, and later on the Eclipse, which has all these features with different fragmentation modes, including also the UVPD, where we can do photon-induced dissociation. And we can get all this information that I just mentioned. So when you consider how many different experimental settings you can have when you want to study all these fragmentation methods, then you have ADC target, you have also the ions can come in different charge states. You want to try multiple charge states usually. ETD reagent targets, it can also be varied, and reaction times and types of fragmentation and so on. So when you begin to look at this and combine the different settings, you may end up with more than 3,000 different combinations, and about 1,900 of those would be meaningful and make sense. So that's basically experiment is to have a computer-based script that generates these 1,800 different combinations and feed them into the mass spectrometer, and then wait for it to do its job and acquire the data. So we built an R-based method called top-downer with some XML, and we got some help from Thermo to help us transfer the data to the mass spectrometer, and then also, of course, retract the data again, and then we have some scripts to go through all the spectra and extract intensity information and assign the fragments. So this is the myoglobin fragmentation map, and as I mentioned before, the sequence is on the y-axis. All the fragment methods is on the x-axis here with different settings here below. So the orange boxes here indicate that it's a C-terminal fragment. The green ones are N-terminal fragments, and the blue purplish ones are where we have both the N and C terminal fragment covered. So just by looking at this, you can see that CID and HCD are not really good for intact mass MSM or intact sequencing of myoglobin. Very few fragments. ETD generates fragments throughout the sequence. ETCID even a bit more. ETHCD also work, but not as well as ETCID in this case. And you can also see that when you increase some of these parameters too much, you end up having no fragments from the middle, but you optimize for the N and C terminal fragments here. So you want to have this compromise setting where you get both the N and C terminal, but also some central sequence fragments from proteins. So we then ask the questions, how can we combine the methods? So we want to have the method that gives most fragments, and then we take the second best method would be the one that complements the first method the best, to add even more fragments, and then the third method would be the one that complements the first two. And when we take the top 10 methods in that way, we actually can get up towards 95 to 99% sequence coverage by combining the best settings here. So that's the whole purpose of this exercise, is to try to find out what are the best settings for proteins, and maybe can we find a few settings that would work for all proteins. Unfortunately, the answer is no. Each protein behaves in a different way, at least so far. But we hope that over time we will get enough information that we can actually learn from this. And you can, of course, imagine analyzing many, many proteins and then have some deep learning method that helps us to optimize for particular proteins. So here's carbonic anhydrase, which is 28 kilodalton protein. We get 85% sequence coverage. And again, you can see the CID and HCD is not working well. We get more info with the ETD and ETCID. But then we need to combine many methods here to get up towards the 95%. But we can do it because the data is there. We just need to find it in the data set. So we can also go in here and look to see which fragments do we see for each method. So for CID, we see Y ions and B ions as expected. Same for HCD. When we move to ETD and ETCID and ETHCD, we see the C and the Z ions. And when we move to the UVPD, we see the A ions as well, which we rarely see in the other cases. So we can extract all this information. And as I said, we have these bubble plots where we also can see which settings are best for each method. But I'm not going to go into detail with that now. We can also do different statistical analysis to see how much a particular method or particular parameter influences the fragmentation, but I'm going to skip that to save some time. So now we're using this method actually to study protein drugs. This is a PhD project by MASSA, who just handed in her thesis last week, actually. So here is the results from filgrastim, which is a 19 kilodalton biological. So it's a protein drug used in the context of cancer chemotherapy. So we are looking at 3 different chart states, and we do a systematic MSMS for each of these 3 chart states. And again, here's the map. And you can see that ETD and ETCID works really well, and also with ETHCD actually. And UVPD actually gives a lot of information as well. So with this protein, we get all these fragments when we look at the top 10 methods among all the hundreds of methods that we are surveying. Very nice fragmentation, lots of C-terminal fragments here. That's the greenest part here in this heat map representation. So many C-terminal ions, which you also can see here in the map. So we get 99% sequence coverage here. And we also can distinguish between the cysteine bonded and the unfolded, denatured, reduced protein in this way. So with this top-downer tool, we can now begin to systematically analyze intact proteins and acquire data and learn from this data. We find that we should use the maximum automatic gain control target in order to get as many ions into the system as possible. These are intact proteins, so they carry lots of charge. So we want to maximize the injection of ions. We also have learned that we need to study different charge states. We shouldn't just go with one charge state like the major one. We should also look at the lower charge states and higher charge states in the distribution. We found that the ETD reagent gas target has little effect on the fragmentation. And by varying the ETD time, we can, tune whether we want the terminal ions from the NNC terminal or we want to go for the core of the protein to get more fragments in the center. And it seems that CID is actually very good in some cases, especially together with ETD rather than HCD. But we are still looking into that, and that may be a bit protein dependent. And so if you only have one method, then ETD with supplementary activation like ETCID would be a nice method. And then UVPD actually turns out to be very beneficial as well in these cases. So we get on the order of 90 to 99% sequence coverage for the proteins we have tried so far. And right now we're looking into some glycoproteins as well to see how we can analyze those and map glycosylation sites with this top-downer script in a systematic fashion. So this is my closing slide. So I hope I have shown you some interesting examples and maybe fascinated you a bit about what you can do with middle-down and top-down proteomics approaches to study proteoforms and this diversity of modifications in proteins. And I'm really excited about the unknowability spectrometry because it provides a new dimension for separating isomeric species and isobaric. That means nearly same mass species. It brings a new dimension, and it's a hot topic now in bottom-up proteomics, how to use this collisional cross-section also as a parameter to identify proteins. And the same would go for the larger peptides in the middle-down approach, and maybe for intact proteins and native protein complexes as well, as has also been shown by colleagues around the world. And of course, for all these experiments, you really need to integrate both the experimental methods, the mass spectrometry, with some clever computational methods and algorithms to get the best out of your data. And I think that we'll see more and more intelligent mass spectrometers in the future, because we get more and more data and we learn from it, and that learning can be put back into the mass spectrometers, so they perform more smart analysis of peptides and proteins. So I also just want to advertise here that right now we have a job opening for a proteomics data scientist in my lab to look at clinical proteomics data. So we also, trying to get more data scientists into the group so that we can expand on the computational efforts in the context of proteomics. So with this, I want to acknowledge all the colleagues here, the PhD students and postdocs who have done the work I've showed you. Andre worked a lot with the Histones, the middle-down approach, and Veit Svemle is helping out a lot with the proteomics informatics, of course. Pavel was running the unmobility and also the top-downer approach that was taken over then by MASSA. I want to acknowledge all our funding sources, of course, my colleagues in the protein research group. These are group photos over the years. Some of you in the audience may recognize yourself. I know we have had many Brazilians visiting over the years. And I also want to thank my family for supporting throughout all these studies. So thanks a lot for your attention.
Thank you very much, Ole, for your wonderful talk. Really, really nice. And I always love the names that you and PR group give for the approach and the method. So the top-downer, for example, it's a really nice one. And also, as a comment, I think Luis is a student that presented a lecture here. He studies mosquitoes, so he probably would like to use this new strategy that you developed to analyze an intact mosquito. I think you would love that. So
thank you. Can you hear me? Because it seems that
yeah.
My photo has frozen right now.
Yeah. Your photo has frozen quite a while ago, but fortunately, it gives it's still with a nice shot for you. So it's fine.
Right.
We have already. That's actually Rafael Melani. He's making a question here. That's the one that I would give it to you. So I'll put here for you. I don't know if you can read it. Otherwise, I can read it for you. Melani is the one that I mentioned to you that he's working in Neil Kelleher's lab. So he's a top-down guy.
Yeah. Good. Yeah, good question. So for this, I assume you talk about the top-downer method that I showed in the end. So we work with these model proteins and pharmaceutical proteins. So we are not really limited with sample, but we do nanospray. So we do rather dilute solutions with 1 to 5 micromolar solutions. So we use very little material we do one injection by nanospray, if you can call it one injection. So basically, you load up some microliters of sample in your nanospray emitter, and then you can measure for hours. So of course, you need to ensure that you have a good spray stability for your nanospray emitter, and then you would usually measure for some hours. Depends a little bit how many settings you want to scan. But anywhere from 1 hour to 24 hours, depending on how much you want to explore for one protein.
Great. I don't know if I can make a question first, and then I'll give my colleagues the time. But do you think, Ole, that in a near future, or maybe not a near future, but do you think that it can be, we can foresee it in a way that all these ion mobility technologies will evolve so much that in the end we are not going to need that much MS2 to identify our data, just based on MS1 and ion mobility time, drift time, things like that?
I think maybe for some known species where you have standards, it may be enough, but it will depend on the complexity of your sample. I like MSMS, so I always, I think, would like this confirmation. But of course, it complicates things and it takes time to do it. So it would be really cool if you could get rid of it, or at least reduce the need for MSMS. But I would rather go upstream and say that maybe unmobility could be used to get rid of the LC part, right? So that you could take your very complex samples and spray directly into an unmobility device and maybe do tandem unmobility or multiple tandem mobility to separate ions in the gas phase, or separate your proteins and peptides in the gas phase before you do the tandem MS. And I mean, it's going in that direction, right? So like David Klemmer and others are looking at these tandem unmobility devices to do very efficient gas phase separation. So I think that's really fascinating, and it may take another 5 to 10 years, but the field is moving very fast now with the unmobility developments. So there's the slim approach, with these very long unmobility cages that Dick Smith has developed. So there's a cyclic IMS and so on. But of course, it all depends on the duty cycle you have in each of these unmobility devices and how you can combine that with the MS and the MSMS experiment.
Yeah,
so in some cases you will get rid of the MSMS. In some cases you cannot.
But I was impressed with how fast it is to make this to increase a lot this drift time, right? To do like this 20 cycles to 60 cycles. It's still below 100 milliseconds, right? So
yeah, it's in the order of 50 to 150 milliseconds. So it's pretty fast. So it's the same you do when you do like an orbit trap experiment. Sometimes you need that time frame to do ETD or other types of MS and MS to the third and so on.
It's faster than ETD fragmentation, for instance, the activation. Yeah, really nice. So I don't know, Giuseppe, Fabio.
Can I ask a question, Joe? So first of all, Ole, thanks a lot for your presentation. I really enjoyed. And I would like thanking you for sharing also this first slide about the PNAS paper, which I had already seen in the LinkedIn. You posted there, so I had already give an okay for that, because it was actually, I think, one of the first papers that showed the potential of proteomics. And still, as you said, it's true what was written there. And so thanks for that. Regarding the top-down R software that you developed, you mentioned at the end that you are already applying for glycoproteins. So actually thinking about a little bit of my project, could you share some information on how these different fragmentation are working on glycoprotein? If you have some something to share with us about that?
Yeah, of course, it's still very early days, but it seems that at least for some proteins, you can very nicely map out the glycosylation sites. And for others, especially for larger proteins, it becomes more difficult, which is no surprise. But then, of course, you can have other complications by other modifications. And if you have disulfide bridges and so on, then it changes the fragmentation behavior and so on. So it's not straightforward. I think that's the honest answer. But we're working around some of these challenges and trying to come up with clever ways to map out modifications in the intact proteins as well.
If I may ask, are you currently working on both N-linked and O-linked glycoprotein, or are you starting with the N-linked with just one side?
Yeah, mostly N-linked. We have a few O-linked as well, and the O-linked are usually smaller glycans. But I mean, we don't have a lot of data yet, so it's still very early days.
Okay, thanks. May I just take for another question? Actually, it's related to something was not part of your lecture, but I mean, you recently published the application of MIPS, molecular imprinted polymers, for histidine phosphorylation. I think they are defined, if I'm not wrong, as a plastic antibody. I would like to ask you, what is your opinion actually for the future application of this MIPS in other kind of PTMs?
Yeah, I think it's another project that is in a very early stage, but it's also a fascinating field, to use polymer chemistry to actually make an imprint of your favorite modification, could be a phosphorylation or histidine phosphorylation, for example. So we have tried with phosphotyrosine previously, and that was published some years back. We saw some enrichment of phosphotyrosines, but we had to use it together with titanium dioxide. The phosphohistidine data actually looks more promising, I would say, and it was also recently published, that we will enrich the phosphohistidines, but we need really to show it for real biological samples. So we have shown it for rather simple mixtures that were mixed in the lab, basically, and we see some enrichment, but when you move to biological samples, it's a different ballgame. It's much more complex, and you have much higher background. So yeah, more development is needed here, but it's a really clever idea, and we are working with the Swedish chemists in Malmö University, and now they're thinking about how to change the chemistry to optimize these MIPS even better, get them to be more specific and selective. So I think, yeah, within some years we will get better at this, but it's still very exploratory and very early days. It's really fundamental science, which is good fun, and yes.
Also,
I think as long as if you can. We have talked about glycans also, and sialic acids and so on, and there's some selectivity, but it's not that convincing yet, I have to say, because the glycans are very bulky, and yeah. It's not easy to design and make such a polymer scaffold that will capture that with high affinity.
Well, MIPS are, some of them are already used, for example, in environmental sciences for actually purifying some contaminants in water.
Yes.
So I really believe that all the optimization that you are contributing for that, I mean, at some point will come that we might substitute antibodies, or maybe even better, we might use them complementary, because we have already seen that on a very complex mixture, applying different enrichment strategies that use different physical-chemical properties, we don't get 100% overlap. And actually, this means that to get the phosphoproteome, or in that case, the sialium, maybe we need to use more than one technique to get the bigger picture of the sample. So I think they might be very nice complementary tools for studying PTMs.
Yeah, I completely agree. At the moment, there's no way they can compete with antibodies, of course, but I hope over time they'll be complementary.
Yes.
And give some. I mean, once we find out how to design these things, then you could make anything, right? And it would be much easier to make such a polymer than to make an antibody, because antibodies are not easy to get in a. With high specificity and avidity and so on. It's not just getting an antibody, right? You need to get the right antibody that has the right specificity.
Exactly. And if I can just add just some thought about this topic we are talking about, maybe it will come up some MIPS that are specific to some isomers, positional isomers of the histone tails. Who knows? Might be something that could be worked out to use MIPS for isolating specific histone tails with specific modification. Maybe we'll come there. But I think
we are actually discussing that, how to take advantage of those MIPS. But I mean, you can also do with antibodies to a certain degree. But of course, with the MIPS, you could also imagine having multiple modifications together, which is very difficult to capture by antibodies.
Yeah. Thanks a lot.
Sure.
Hi, Ole. Okay, thanks for the excellent talk. Really great to see your work and all the applications that you have done with mass spec in proteomics. Also, I enjoy a lot the picture of the cover PNS from the PNS paper. Actually, I remember me when I went to Denmark with a lot of protein spots from 2D gels. That's really, really nice. But yeah, I have just one question. Of course, it is in top-down proteomics, and actually, I think that one of the big problems is the interface between the sample and the mass spec. How do you think we can can talk something about that? How can we fractionate the proteins to insert them in the mass spec, something like that? We have a work in this kind of we are doing kind of application in this field.
Are you're talking about top-down approaches or?
Yes, top-down.
Yeah. Yeah, I think it would be very nice to have some new technologies to separate proteins in a way that they could then be fed into the mass spectrometer. I mean, of course, LCMS is the obvious one. It works for many proteins. It doesn't work for some types of proteins either, unless you change the solvents and so on. So that's the more straightforward approach. That's a proven approach with the pros and cons. And now capillary electrophoresis is also, let's say, coming back. It's like 25 years ago, there was a lot of excitement about CEMS, also for peptide and protein analysis, and then it went away a little bit, at least in academic research. I think it still has it's a big thing in pharma industry. But now it's coming back also for proteomics, both for bottom-up proteomics and also top-down proteomics, and interfaces are improving. I still think the commercial interfaces are not that great. Not that I have a lot of experience with it, but I think with further optimization, then CEMS will be really a extremely interesting method to work on. But as I said, also, why not get rid of this separation in the liquid phase and then use ion mobility instead, and just take your protein extract and nanospray it, the whole mixture, and then separate by ion mobility. Of course, there would be some ion suppression issues to cope with, but maybe some simple fractionation could then be used up front, or some very brute force chromatography. So it is a big issue, and I think with the mass spectrometers being so advanced as they are now, I think more and more effort will go back to the front end and how to optimize the sample preparation, and we need to use all our skills and imagination in protein chemistry to come up with clever ways. I think in the future.
Yeah. Thank you.
Yeah, Gilberto, do you want to say something? Your mic is mute. Okay, I don't know. So yeah, Ole, I don't know. Actually, I don't know if I can ask you that, because I've heard that you have just stolen the timsTOF in the lab, right? Or a few months ago, I don't know. And we do that. One of our sponsors is a NobiTrap vendor. But I'd like to hear from you. What do you think about the instrument? What are you planning about that? If you are going to use the timsTOF, because as far as I remember from your slides, you have the MALDI 2.1, right? The one that you can also mount a MALDI source in front of it, right? So are you planning to use it more for imaging and also top-down to use the ion mobility, or you are already using it routinely for bottom-up proteomics? I'd like you to comment about the instrument.
So we actually got two timsTOF instruments earlier this year, but our use of it has been a bit delayed because we couldn't get the training, because the Bruker colleagues, they could not travel due to the COVID situation. So we only recently got the let's say the deep training in the system, but we of course have used it already. So we have one timsTOF Pro 2, which is the sort of the proteomics machine, and it seems to work very nicely. We have not I mean, we don't have enough experience yet, but I mean, it compares very nicely to our Orbitrap systems with sensitivity and so on. We are exploring the DIA passive approach for quantitative proteomics. We are playing with the TIMS as well to look at proteins to see if we can take advantage of that type of unmobility. On the other instrument, that's timsTOF Flex, which has both the electrospray source and the MALDI source, as you said. And the MALDI source has this post-ionization, so it has 2 lasers, basically. So we're going to just play with that, for imaging purposes, but also for regular MALDI, because for some types of biomolecules, especially lipids, this MALDI-2 should give a boost in sensitivity, but it's too early we don't have any data on that yet. We're still just playing around to get to know the system. So it opens some new doors, to explore various new approaches to study biomolecules. So but for imaging, I'm pretty excited because this timsTOF Flex is really, really fast. It has a 10 kilohertz laser, and it just blazes over this tissue sections. So what took 10, 12 hours on the old imaging instrument now takes less than 1 hour. So the speed really makes a difference on that system. So we'll get much more imaging in the future.
And do you have any device to spray the matrix? Because I mean, I never done multi-image in my life, but I've read that one of the most challenging part is to cover it nicely and evenly, right? With the matrix. So do you have any device to do that for you?
Yeah. We have a device called an iMatrix sprayer. It is kind of an open source sprayer. So it's a small robot that you can buy from this small company in Switzerland. It's not very expensive. It's on the order of $8,000-9,000 US, which is 1/10 of the price of some other vendors. And it works fine, but it's of course not as advanced as the more expensive competitors. But it does the job, and all the data we have so far, we're happy with, and we also published based on that. So it's a good way to get started using this iMatrix sprayer. And then, of course, you can upgrade later on when you gain more experience and have more special needs and funding.
Well, I don't know if any of you have more questions. People here in the chat, they are also congratulating you, and yeah, that was a really wonderful talk, everybody says. And I don't know if you've seen here, but Simone was also in the audience, so he said hello to you and excellent work.
I saw his little greetings, but I don't see the chat, so people have to speak if they have, or write if they have some questions.
Yeah, exactly. Yeah. Okay, so Ole, thank you very, very much. It was really, really nice for us to have you here. Thank you for accepting the invitation. Merry Christmas for you. Happy New Year for all of you, and I really hope that I can see you all in person in Denmark or elsewhere soon.
Yeah, I certainly hope to see all of you again. It was great to see you online. Many good friends in Brazil and around the world. So Merry Christmas to all of you, and thanks again for the invitation to give this talk online. It's amazing that we can do this, across the world, and
exactly.
It's almost like sitting next to each other, right?
Thank you all.
Yes, thanks a lot.
Greetings. Cheers.
Yes. Bye-bye.
Bye.
Bye-bye, Ole.
Thank you.
So, agora tá com você, né, Gilberto? Estamos acabando aí a sessão, né?
Vê se ainda tem gente aí, né? Ainda tem espectador.
Eu ainda não soltei a lista de chamada.
No início eu fiz... No início eu enfatizei, ou reenfatizei, o que eu havia dito no último seminário sobre as eleições da para a diretoria da BrProt, né? Eu queria renovar agora, nesse final, a participação de todos na eleição da BrProt. Pensem bastante, botem suas chapas, concorram à diretoria, né? E vamos tocar a BrProt renovada. Nós temos um calendário que foi enviado para vocês, tem aí coisa de 1 mês para a montagem das chapas. Eu sei que é Natal, ano bom, mas também confraterniza profissionalmente, não só familiarmente. Então, a gente pode montar uma chapa, mandar, o conselho diretor vai analisar as chapas e a eleição se segue dentro do calendário, e depois a gente tem a Assembleia Geral para homologar essa eleição e passar adiante a diretoria da sociedade. Vocês veem que a proteômica está num crescimento extraordinário. A participação, o comprometimento de todos no desenvolvimento da proteômica no Brasil. O Brasil é o líder da América Latina em proteômica. Então, e quando eu digo líder, é muito à frente dos outros também, graças ao trabalho de todos. A gente tem que manter isso, tem que não só manter, como desenvolver mais. A gente está fazendo tudo que pode para trazer excelência para vocês, mas uma nova diretoria será muito bem-vinda, com novas ideias, nova dinâmica, novo comprometimento, sangue novo, etc., para tocar a nossa BrProt. Inclusive para o congresso do ano que vem, conjunto com a MASSA. Vamos explodir nesse congresso. As perspectivas são extraordinárias, trazer gente de altíssimo nível para seminários, a BrProt vai ter uma participação muito ativa e a nova diretoria vai ter que trabalhar muito nessa direção. Era só isso, e agradecer a todos, né? Viu, Tiago? Agradecer a organização, vocês, né? Fábio, Magno, Giuseppe, Ana, Luiz, Jéssica, todos os alunos que trabalharam, não quero esquecer nenhum nome, e vamos voltar ano que vem. Ano que vem vai ter mais e melhor ainda. Mais renovado, melhores prêmios, maiores premiações e um Feliz Natal para todos.
É isso aí. Gilberto, agradecer também a Analítica, né, que tem já aí a segunda edição, que eles têm ajudado a gente demais, né?
Isso. Eu esqueci.
Bom, e aí temos que falar agora também do resultado, né, das 2 sessões que nós tivemos, né? Vamos começar primeiro falando da sessão dos Student Section. Eu estou pensando, não sei o que vocês acham, mas eu pensei em colocar aqui, transmitir como é que ficou aqui o resultado, como é que foi feita essa seleção. Vocês acham que é ok ou não? Porque o que é importante a gente explicar? E aí fica, na verdade, também uma percepção nossa, né? E aí fazer um pedido, né, para que mais grupos participem do My Friends Massa. Isso aqui é feito para todo mundo, né? A gente quer que mais pessoas possíveis do Brasil participem, enviem trabalho. Nessa edição especificamente, muitos alunos dos organizadores enviaram resumos e não tanto dos demais grupos de proteômica do Brasil. Então, a gente queria, de novo, pedir, por favor, para vocês enviarem. É muito importante para a gente que isso aqui seja, né, mais diversificado possível. A ideia é que isso seja um evento da proteômica nacional. Então, por favor, participem. Para o próximo evento, talvez nós vamos pensar em novas formas, né, de tentar aumentar o engajamento de outros grupos, né, para se sentirem mais pertencentes ao evento, e nós vamos tentar trazer novas ideias. Então, falando isso, eu acho importante também, né, em favor da transparência, mostrar como é que a gente fez essa seleção. Então, nós tivemos, né, agora eu estou na dúvida se foram 10 ou 9 resumos submetidos para a sessão de estudantes. E como que nós fizemos? A gente enviou esses resumos e também, depois que os resumos foram selecionados, pedimos para essa comissão avaliadora assistisse todas as palestras e quem ficou, então, responsável pela avaliação foi basicamente o conselho da BrProt, juntamente com a direção da BrProt, tá? E como é que a gente fez? Por exemplo, eu tive um aluno meu que ia apresentar. Então, eu não avaliei o trabalho dele, e isso foi feito com todos os estudantes. Então, o Giuseppe também teve uma aluna, teve 2 alunas que vieram ao trabalho, acho que foram 2 ou 3, agora estou na dúvida. Então, ele não participava da avaliação. Se tivesse alguém também do conselho que participasse do trabalho também, não avaliava. E depois essas notas foram normalizadas pelo número de pessoas que poderiam votar naquela nota. Então, aqui eu estou só chamando aqui, mostrar rapidinho. Então, basicamente, como é que era feito? Cada um dessas... nós tivemos só uma pessoa que não pôde enviar a nota, mas aí cada avaliador escolheu posição de 1 a 3, e esses valores, cada posição tinha uma nota. E aí depois tinha, então, a somatória das notas, né? Você tinha, então, aí toda a média, a somatória de todas as notas que aquela pessoa ganhou. Por exemplo, a Janaína, que foi a mais bem votada, ela teve 5 primeiros, uma de indicação de primeiro e 1 de terceiro. Após, então, multiplicar por esses valores aqui que nós demos arbitrariamente, era depois dividido pelo número de indicações que ela teve. Isso para evitar que estudantes que tivessem menos pessoas avaliando, porque mais pessoas que estavam avaliando o grupo participavam do trabalho, não fosse prejudicado. Então, dessa forma, eu acho que a gente conseguiu mais ou menos ser justo. Então, a gente teve que, em primeiro lugar, é importante falar, gente, que todos os trabalhos que foram apresentados e os que não foram selecionados, os resumos estavam muito bons mesmo. Foi um consenso geral que foi muito difícil escolher os melhores trabalhos. Mas, então, nesse sentido, a Janaína, né, que, na minha opinião, foi um trabalho belíssimo, eu acredito que de todos, né, foi, então, o primeiro lugar. Ela vai ganhar, né, um certificado do prêmio, né, do melhor trabalho apresentado no Student Section. E aí depois a gente teve aí em segundo lugar e terceiro lugar o Lucas e a Jéssica, que vão, então, receber também uma menção honrosa, tá bom? Então, parabéns para os ganhadores e parabéns para todos os que apresentaram. E, por favor, mandem mais trabalho, tá, gente? A gente tem certeza que tem muito mais trabalho de proteômica sendo feito no Brasil, então, por favor, considerem enviar o trabalho para a próxima edição, tá? Com relação ao Young Investigator, na verdade, foi a primeira vez que nós fizemos nós tivemos a ideia de fazer essa sessão. Eu acho que foi uma ideia muito boa, porque tem vários pós-docs e recém-doutores aí que fazem trabalhos fenomenais. A questão é que a gente sentiu que ficou faltando uma coisa que depois a gente a gente vai aprendendo, né? A gente vai aprendendo com o tempo e aprimorando. Então, o que a gente sentiu é que, no final das contas, a gente deixou muito aberto, e aí acaba sendo muito heterogêneo, né? Então, tinham trabalhos que eram só feitos 100% no Brasil, trabalhos feitos 100% no exterior, trabalhos que foram realizados metade aqui, metade lá, em diferentes tempos, né, de doutoramento, né, de cada um deles, tá, obviamente, diferentes estágios da carreira. Então, assim, como a gente não definiu nenhuma regra, a gente achou que seria muito injusto a gente falar que, óbvio, que a gente avaliou, tiveram uns que nós gostamos mais ou menos, mas todos foram excelentes, né? A gente teve um trabalho que usou várias estratégias de proteômica estrutural para caracterizar uma determinada proteína, estratégias na fronteira do conhecimento que estão sendo desenvolvidas para a gente poder, então, depois conseguir ter uma boa caracterização, né, de proteínas como anticorpos, e trabalho que utilizou a proteômica como uma geradora de hipóteses e depois validou e teve, assim, um trabalho super fechado. Então, para não cometer nenhuma injustiça, nós definimos que os 3 vão receber o prêmio. Então, parabéns, Viviane, Rafael Melani, Gabriela, vocês 3 vão receber, então, o prêmio do Young Investigator Award. E, como eu falei, para o próximo, a gente vai aprimorando e vendo como é que a gente faz isso da forma mais justa possível. Tá bom? Então, parabéns para todo mundo. Parabéns aí, né, para a Janaína, ela está aqui agradecendo. Parabéns, Janaína, esse trabalho sensacional. Você está indo aí num caminho brilhante. Parabéns também para a Jéssica, parabéns para o Lucas, mas parabéns também para o Luiz, para o Maurício, para a Alicia e para todos os outros que enviaram o trabalho. E, mais uma vez, gente, mandem trabalho para o próximo evento. Vou abrir aqui a palavra, não sei se o Gilberto, o Fábio, o Giuseppe querem falar alguma coisa.
Só agradecer novamente, foi um ciclo, né? Esse segundo ciclo de 2000 é o terceiro ciclo do My Friends MASSA e o segundo desse ano, então realmente sempre dá um trabalho no início, mas depois que a carruagem anda, é muito prazerosa, né? Você está aqui toda semana, né? E ver os trabalhos de excelência e estar com amigos, né? A gente vê aqui no YouTube todo mundo comentando, né? A gente sempre vê as caras amigas aí e as palavras amigas do pessoal. Então, assim, obrigado a todos que conseguiram assistir, participaram desse ciclo de seminários. Desejo a todos também boas festas de fim de ano. Então, um abraço a todos e a gente se vê no ano que vem, né?
Tiago, eu queria só reiterar o que você disse. Eu acho que a participação está muito baixa. E isso não é legal. Quer dizer, todos podem concorrer. Façam bem os seus trabalhos. Vocês têm excelentes orientadores, têm excelentes teses, excelentes trabalhos. De todos os níveis podem participar. Então, vamos aumentar, vamos incrementar isso, vamos ter uma competição saudável, de excelência, em que vocês mostram os seus trabalhos para os colegas. Isso é muito importante profissionalmente. Vocês têm que aparecer, o seminário é uma tela para mostrar o trabalho. Então, para a comunidade, né? Por favor, participem, entrem na disputa, entrem na competição, entrem na brincadeira, mesmo sem competir. Mostre o seu trabalho, traga o seu trabalho para a avaliação. A avaliação é uma coisa importante. Passar na avaliação e apresentar o trabalho é uma medida da excelência do seu trabalho. E se você não entrou, tem que melhorar o seu trabalho para entrar da próxima vez, mudar o nível do trabalho e melhorar. Então, a participação de todos é fundamental para que a gente tenha uma melhor excelência nos seminários. E o comprometimento, então, eu nem digo que é fundamental também. Feliz Natal a todos e obrigado a todos. Renovo meus agradecimentos, inclusive à nova analítica, que o Tiago lembrou, né? Sem a nova analítica, a gente não poderia estar dando esses prêmios. Uma parceira muito forte da gente.
Posso só adicionar, na verdade, reforçar as palavras do Fábio, do Tiago e do Gilberto, parabenizando não só os primeiros 3 que ganharam, mas também todos os alunos que apresentaram. Foram, assim, apresentações excelentes. A gente com certeza vai repetir isso. Eu acho que é um senso comum que a gente quer que isso até se estenda, como o Gilberto e o Tiago falaram, que precisamos de mais pessoas. Mas, assim, eu queria agradecer aqui o Gilberto, o Tiago, o Fábio, o Magno e todos os alunos da organização, porque, assim, a gente organizou 2 My Friends MASSA durante a pandemia. Eu acho que foi uma forma para a gente continuar a demonstrar que a proteômica no Brasil está viva, é bem representada por todos os alunos, todos os jovens pesquisadores e também os palestrantes que contribuíram para esse evento. Então, aí vão meu agradecimento para toda a comissão e também os alunos que contribuíram para isso. Obrigado.
Beleza, gente. Então, a gente encerra aqui a terceira edição do My Friends MASSA. Esperamos, então, estar firme e forte ano que vem para fazer a 4ª edição, né? Com mais resumos. A gente quer trabalho para avaliar esses resumos. Então, mais gente enviando resumo. Muito obrigado.
Vou jogar uma ideia aqui. Quem sabe aí no futuro a gente faz um simpósio do My Friends MASSA presencial, né?
Exato. Angra dos Reis, né? Algum lugar assim.
Búzios.
Eu acho que dá, viu? Eu acho que dá. Gente, tchau. Pode falar.
Não, valeu, era isso. Era isso, Tiagão, valeu.
Abraço, viu, gente? Feliz
Abraço.
Natal, feliz Ano Novo para todo mundo e a gente vai se falando aí.
Feliz Natal.
Beijo para todos. Tchau,
Boa
tchau,
festa,
gente.
pessoal. Tchau, tchau.